Hear my hopefully inspired introduction. All right, well, welcome everybody. Thank you all for coming to near Dr. David Wirth's defense here. So a brief introduction. I know you're all here to hear what he has to say rather than what I have to say, but well, it's my last time to give him a little bit of hell, so I'll do my best. So David is a local product. Grew up in San Diego, went to high school in San Diego. He has a bit of a unique background coming to the PhD program, which I won't tell you much about because he has some slides detailing what he's done, but you'll see it's a bit of a different path, and I think it's a very productive path. He went to UCLA, go Bruins, got a degree in mechanical and aerospace engineering, BSMS, I believe, and then he went off to do other things, which he'll tell you about, which are all very, very interesting. And then I met him, I think, in 2018, right? 2017 or 2018, something around there. And he's kind of his background as a student is very different than the types of students I take. So mechanical and aerospace engineering, spent time in the military. And so weren't necessarily sure how well we would be able to communicate because we both speak very different languages. But I think we've sorted this out over the past 4 years. And so one of the things that David and I first started talking about when he joined was this idea of kind of living materials and this idea of going beyond the kind of scope of manufacturing, right? Using living types of things as a manufacturing platform, and that kind of spurred the idea of which led into the UCSD MRSEC. So that spun into some $20 million of funding and coming to UCSD. So that's not so bad, I would say. In any case, so David's only been here for 4 years. He's gotten most of his time here was during the heart of the pandemic. However, he's published, well, we'll say 4 papers. Two first author papers have been accepted. 2 are are under review right now. He's got an additional 5 co-authored papers and 1 more first author paper that should be out hopefully in the next couple of months. So very productive in the course of 4 years. I'll stop talking. Everybody wants to hear what he has to do, but welcome, David. We're looking forward to hearing it.
Thank you, John. Appreciate it. Well, thank you all for coming here today. Thank you to the committee for coming here and for all your mentorship and support over the years. Without further ado, today I'd like to present to you my doctoral work on novel methods and materials for additive manufacturing. So briefly today, I'm going to give you a personal background of how I got here, how I got interested in the subject, some of the theoretical work that I've done underpinning the subject area, and then get into the applied research I've conducted here at UCSD. Yes? Oh, the screen's not shared? All right, let me try that. Good call. Let's see if this works. All right.
You're good.
It's getting a little complicated. All right. Yeah, here we go. Oh no. Computers becoming very interesting. All right. So sorry about that brief introduction. All right. There we go. All right. So after that little debacle, I'd like to talk a little bit about my personal background and how I got involved in this field. So before I came to John's group, I graduated with a master's and a bachelor's in aerospace engineering from UCLA in 2012. Following graduation, I commissioned as an officer in the United States Air Force. I mostly worked in Dayton, Ohio. I worked at the National Air and Space Intelligence Center and the Air Force Research Lab. And it was during this time that the 3D printing revolution was really taking off. The Stratasys patents were expiring. MakerBot was just starting to put out their commercial line of 3D printers. and metal additive manufacturing was just rising to prominence. And as an aerospace engineer by trade, I was really interested in the metal additive manufacturing. And so I went to the branch chief of the metal additive manufacturing branch at AFRL, and I asked whether they had a metal 3D printer I could come and take a look at. And the response was a little bit surprising. They said there were no metal 3D printers in the entire Air Force at this time because they were just so large and so complex. And it got me thinking if there was a better way to do things. So I got to tinkering around in my garage, and this led me to the field of localized electrodeposition and electroplating. And over the course of a few months, I put together a prototype of a metal 3D printer that operated at room temperature under ambient conditions. After this discovery, I partnered with some of my colleagues from San Diego to found a company called Fabricate Labs. Using some MEMS technology, thin film transistors, and some more advanced circuitry, we were able to significantly improve the fidelity of these metal 3D prints at room temperature. So where this is going is I was able to successfully bring a novel subject field in to enhance the capabilities of additive manufacturing. And it got me thinking about what other subject fields can we leverage in order to enable new possibilities in the field of manufacturing. Now, before I talk about these possibilities, I want to talk about manufacturing as a whole, and specifically whether our current definition of manufacturing can be more broadly defined. So normally when people think of manufacturing, you might think of milling a part, machining brass, or printing plastic. We can bifurcate the field of manufacturing into additive versus subtractive by thinking about where the material is going. But it's perhaps more interesting to think of this field as a question of where the material is going and also where is the information coming from. Manufacturing as a whole can be thought of the conversion of digital information into physical structures in a granular fashion. And using this definition, this allows us to look at the field of synthetic biology and directed assembly as manufacturing systems. Now, the terms top-down and bottom-up have pretty rigorous definitions in the field of nanotechnology, but it's my assertion they can be applied to manufacturing as well. So when I say top-down manufacturing, I'm saying manufacturing processes where information is coming from an external assembler, which encompasses the part that's being formed and builds a structure at a linear or a polynomial rate. And the total volume of this part is limited to that enclosed by the assembler. It has a build volume. With bottom-up manufacturing, there is no build volume. The assemblers are mobile, and they're smaller than the overall structure which is being built. And if the assembler is self-replicating, it can assemble structures at a logarithmic rate rather than a linear or polynomial rate. Now, there's been a lot of work done on expanding the field of manufacturing as a whole, but what I really wanted to ask in my theoretical work is what is the limitations of manufacturing? In other words, what is the fastest that a given manufacturing system can produce a part, given these physical constraints and the resolution of this part? So to answer this question, I'd like to look at a notional top-down additive manufacturing system. So in 2017, Maxim Schuster published his dissertation at MIT on volumetric additive manufacturing. And in this dissertation, he published a graph where he plotted a number of different additive manufacturing systems with a number of different technologies, color-coded biotechnology, plotted on a graph of material deposition rate versus resolution. Right? And he just kind of put this graph out there and he didn't say that much about it. But if you notice, there is a correlation. All of these incredibly different systems fall onto these neat, relatively neat trend lines. And if you look into these individual data points, you'll see that you can move along the trend lines just by varying the size of the print head. And you can move diagonal by increasing the complexity of the system. In other words, increasing the number of simultaneous deposition paths or the speed at which these deposition paths are moving. So we can rearrange these same points on a plot of minimum feature size versus build rate to get a positive slope. And we see that this slope on a log-log plot is roughly 3. So now the question is, why is it 3? Where does the 3 come from? And why is there this correlation between all these different types of manufacturing systems? And can we elucidate this? Can we elucidate where the slope of 3 is coming from and what goes into the y-intercept? So we can do this by looking at a notional additive manufacturing system, which is just an FDM nozzle moving along a vector, and this nozzle can turn on or off in response to digital instructions. Now, the absolute maximum rate that this printer can build an object is going to be with the nozzle always on. So using some more geometric constants of this system, in other words, the velocity that the nozzle is moving at, the layer height, radius of the nozzle, et cetera, we can develop an expression for the volumetric build rate as a function of these known geometric constants. And this relationship is somewhat agnostic of the type of printer you're looking at, right? This can apply to FDM or SLA or even multiple highly parallelized systems such as masked SLA or continuous liquid interface production. Just by adding this factor of n, which is the number of simultaneous deposition paths. And so using this, we can come up with a relationship for the volume or the time to print an object with a known volume, which is related to the volume of the object, the resolution you're producing at, and these known geometric constants. Rearranging this relationship and taking the log of both sides, we can come up with a relationship in the form of y equals mx plus b. And we see that the slope of 3 is coming from the cubic relationship of minimum feature size. Furthermore, we can see that the y-intercept of this plot is coming from the number of simultaneous deposition paths and some constant representing the geometry and the speed of the printer. So now that we have this notional relationship, the question is, can we push the envelope? Can we use what we know about manufacturing systems as a whole, and novel methods and materials, to produce systems which break free of this standard envelope of manufacturing? So this leads me into my applied research here at UCSD. And the first topic I'd like to discuss is harnessing isotropic expansion to enable more effective additive manufacturing in resource-constrained environments. Now, when I talk about resource-constrained environments, as an aerospace engineer by trade, I've always been interested in materials and processes which would allow future explorers to survive and thrive beyond the bounds of this planet, or space habitation, Mars colonization, etc. But some of the most useful structures for this application won't fit on the end of a rocket. They have to be deflated or compressed in some way and assembled at least partially from information on site. So in the olden days, a settler or explorer coming to a foreign land, they wouldn't bring their house on the back of a wagon. They would bring digital information in the form of plans to build a house in their head, and they'd bring a tool for this assembly, an axe, and they'd use the resources at their destination to construct an object larger than they could transport. Now, unfortunately, there's no trees on Mars, but maybe we can develop some better materials and better methods for explorers to craft tents in the future. So going back to this relationship, what are we trying to do? Well, we're trying to make structures which would ordinarily require a very large assembly system that might be very difficult to transport using a small assembler, and then expand them to make these larger structures. So the ideal material for this application is compact and simple during transport and large and complex on site. So in this example, I show an FDM filament that you're printing using a small 3D printer, and you expand it to make a larger part. There's already a FDM filament with these sorts of properties. It's called ColorFab Lightweight Foam PLA, and it's an FDM filament with a blowing agent that's incorporated into the filament, and it expands upon heating. Unfortunately, FDM is a highly anisotropic material process. In other words, the mechanical properties in the X and Y dimensions of an FDM printed part are different than the Z dimension. So if you were to print something at small scale using this filament with blowing agent, upon expansion, it would have a different geometry than the original part. So really, we want a manufacturing process that's more isotropic. So resin-based processes are much more isotropic. The X and Y mechanical properties, the bonding between the layers is nearly identical to the X and Y bonding. However, this presents a chemistry challenge. Developing a SLA or masked SLA print resin is a lot more challenging than just incorporating a blowing agent into an FDM resin. And this is because in a resin, you can have a relatively homogeneous mixture of blowing agent and polymer because it's just, it's solidified in place once the polymer is hardened into a filament. With a resin, if this blowing agent isn't fully soluble, it'll settle out during the course of long prints. So there's been some prior art in this area. In 2019, Wegner et al. Developed a relatively nozzle hydrazine-based blowing agent, which was soluble in a proprietary sardimer monomer. And he was able to print this resin using a polyjet machine, which is a relatively large, very expensive and specialized 3D printing system. And he was able to expand it using near infrared light. So the challenge here is, can we come up with a blowing agent that's a little bit more economical? Can we use a system that's a little bit smaller, maybe a commercially available system? And can we increase the expansion ratio? So just to reiterate the requirements for this novel foam, we want something that can cure quickly at low light intensity, because commercial mass SLA systems typically have a little bit lower UV light intensity. We need a blowing agent which is soluble in the neat monomer, and we need this blowing agent to decompose between the glass transition temperature and the melting point of the polymerized resin. If it decomposes too early, the polymer is not going to be compliant enough to expand. And if it decomposes too late, you're going to lose all that structural information that was imparted during printing. Finally, we want a resin with a low cross-linker concentration. Cross-links increase the glass transition temperature and reduce the free volume of the polymer, thus reducing expansion. So the final resin that we came up with was a base of 2-hydroxyethyl methacrylate, also known as HEMA. And the blowing agent we chose was diterbutyl dicarbonate, also known as Bach anhydride. Now, this is a non-traditional blowing agent. It's typically used for Bach protection of reactive amines. But in fact, if it's heated above 150 Celsius, it decomposes into 4 moles of gaseous products. And it's fully soluble in the neat 2-hydroxyethyl methacrylate monomer. We also use 2.5% each of these industry standard near ultraviolet photoinitiators. So here's the overall process flow. We create a model using a computer, print it in the small scale using a commercial mass SLA printer, and then expand it using an oven set to 200 Celsius. Using this method, we can create objects up to 40 times larger than the original structures by volume, and create objects larger than the printer's original build volume. So this is a big achievement. These complex geometries hold their shape and size even after removing from the oven. So here's a video of one of these polymer spheres expanding. You can see that an expansion front starts on the part, and because the decomposition of Bach anhydride is an exothermic process, this expansion front propagates until the entire part has reached its final resultant size. So this expansion can be controlled digitally. In other words, just by varying the exposure time and layer thickness of these 3D printed structures, we can control exactly how much they expand. So this is a contour plot of the exposure time and layer thickness, and then the color on this contour plot goes from blue to red, blue being basically 0% expansion, and red being 4,000% expansion. If you go outside of this contour plot, you end up with structures that are not printable. In other words, they're overexposed or they're underexposed. Furthermore, by using very thin layer thicknesses, you can actually switch this foam from an open cell structure to a closed cell structure. And here's a demonstration of this effect. So all of these 1 centimeter by 1 centimeter sample cylinders were printed with the exact same chemical composition, except this front wall. This is control. But you can see this row 2, they're puffing up like marshmallows, and in the back they're expanding maybe 200%, and then the second row maybe 150%. And again, these are all the exact same chemical composition. We're just varying the print parameters. So there's been some commercial interest in this technology. We published a paper back in 2020. In 2022, Desktop Metal released a type of polyurethane-based expanding foam, which they call free foam. You notice that they're also using this lattice or Voronoi structure. Now, these are computer-created patterns where each of the structural members has a roughly equal cross-sectional area. This is important because while the expansion is relatively isotropic, there is still some geometric dependencies. So if I were to print my arm in this isotropic expanding foam, my fist would still expand disproportionately to my wrist because my fist has a larger cross-sectional area. There's going to be more gas trapping than a smaller member like my wrist. But using a Voronoi or a lattice structure, you can create structures which have roughly identical cross-sectional area for every structural member and obtain a very even expansion. So how does this improve the state of the art? How does this relate to volumetric prints? Well, if we look at how long it takes to print a 1-inch sample cube using FDM, it takes about 1.7 hours per cube. Using a DLP or a mass SLA process, we can cut this down to 0.7 hours per sample. Now, this is all at 150 micron layer height. If we use a isotropic expanding foam process, we can get the effective print time for a 1-inch sample cube down to 37 seconds per sample. And this is because the samples are much smaller, so we can print them much faster, and using a highly parallelized process, we can fit many more of them onto the build plate. So what does this mean? Well, it means that we can produce volumetric build rates up to 1/10 of a cubic meter per hour using a system that costs less than $500, and where the system itself is less than 1/10 of a cubic meter. So this is a major improvement over the state of the art. Now, there are still some limitations to this technology. The foams are relatively mechanically weak. It's about the same mechanical properties as styrofoam. And there is, as I said earlier, a bit of geometric anisotropy during expansion. So the next question is, can we remedy some of these limitations? Can we improve the mechanical properties of these foams? So the method we've come up to do this is a two-part approach. So we print the structures at small scale, thermally expand them, and then spray them with an ultraviolet cured photocurable high toughness resin, and then we arrive at a functional part with a little bit higher strength. So there's already some commercial resins with relatively good mechanical properties. Soraya Blue is one of these resins. It's a urethane acrylate based oligomer. Unfortunately, the commercial resins tend to have very high viscosity. So you need a specialized paint sprayer, generally a high pressure, very large, bulky paint sprayer to apply these resins. And this kind of contradicts with the overall goals here. The goals is to reduce the weight of the assemblers. So to do this, we can use if we can find a resin that has very low viscosity, we can use a Venturi-based paint sprayer, which is much lighter. So we came up with a resin that fulfills these properties. We called it S100 resin because it's the 100th resin we tested. It's a isocyanuric acid bisphenol A acrylate-based high toughness resin, boasts 25% higher tensile strength and 50% higher toughness than Soraya Blue. And most importantly, it has low viscosity, so it can be applied with an ordinary cordless paint sprayer. So if you look here, the mechanical testing results of this resin, as printed tensile testing dog bones, you can see a bit higher toughness than the baseline Soraya Blue. And if you post bake it, you can get drastically higher tensile strength. Pulling this thread a little bit and exploring different post bake temperatures, you can actually tune the mechanical properties of this resin by adjusting the post bake temperature. So if you want a high toughness resin, you can post bake between 80 and 120 C. If you want a high strength resin at the expense of a bit of brittleness, you can post bake between 150 and 120. So next step was to do a bit more rigorous testing on the full up foam composites using these 1-inch sample cubes created out of foam. So we printed these, expanded them, and created 4 groups of samples. Group 1 was the baseline foam with no coating, control sample. Group 2 was coated using the S100 resin and then cured using an artificial UV light source. Group 3 was coated and then cured using sunlight, and group 4 was sprayed in the sunlight. So the results from this study, we achieved a 400% improvement in compressive yield strength using just a single coat of this high strength resin. Unfortunately, the density also increased once these sample cubes are sprayed. So the overall strength to weight ratio, or the specific strength, there's not much difference. There's about a 20% improvement for groups 2 and 3 of the specific strength, which is still not, it's still relatively significant, but it's a lot lower than the improvement in compressive yield strength, which is between 330 to 430% improvement. So using this new method, using this new high strength resin, we can begin to create functional structures. So the applied technology demonstration we chose for this was to create a foam drone. So you can see all the components for this drone fit on a single build plate of the Anycubic photon. Here's what they look like as printed, and then after expansion, you can see what they look like. After they're glued together with the S100 resin, you can see that they're dramatically larger than the original build plate. The motor mounts were actually printed using conventional mass SLA printing because they required a little bit higher tolerances. Here's the coating procedure. You can see you just spray them with an ordinary cordless paint sprayer and then cure them using a UV light source. And here's the final drone structure. You can see how much larger that is than the original Photons build plate. It's important to consider that all the structural members for that drone were printed on a single build plate. Here's what it looks like with all the electronics inside. And then here's the drone with all the propellers on. And so the flight test for this, we basically put it, rested on a bush as a launch pad, started it up, and gets about an inch off the ground. Unfortunately, it was structurally sound, but it wasn't weight and balanced properly. So about a second after it gets off the ground, it tips over, catches on a twig, motor falls off, and that's the end of the flight test. Nonetheless, this is the most altitude ever achieved by an isotropically expanded high-strength foam structure. So I would say there's some level of success in this flight test. So for future work, obviously making a more mechanically robust coating, improving the weight and balance of our foam drones, and the RF initiated expansion of these foams. So if you think closely about this process, yes, we are breaking free of the build envelope of our assembly system. However, we're just trading that for the volume of an oven, right? Because these things require an oven in order to expand. So if you had an RF initiated expansion, you wouldn't need to rely upon the build envelope of an oven. So the next, stepping sideways, the next topic that I want to explore is, can we apply additive manufacturing components and subtractively manufactured structures to explore new applications in biomedicine? So injection molding and polymer melt processing are manufacturing processes that are largely free from the resolution, size, and speed trade-offs of top-down additive manufacturing assemblers. Because we're merely copying the physical structure of a mold onto a physical object, there's no conversion of digital to physical information here. So we can produce high-resolution structures much quickly and in mass quantities, which we can't do with traditional top-down additive manufacturing systems. So in this work, we seek to miniaturize polymer melt processing systems to enable researchers to experiment with this manufacturing method for the production of novel dissolving biopolymer implants and medical devices. So the first step here was to see if we can utilize existing FDM hot end technology to create a polymer melt processing system with a little bit better temperature stability. So we succeeded at that and published a paper on that effect. And then we conducted some modeling and simulation work to assess the shear rate of polymers melt processed using this system, and created some preliminary tensile testing dog bones to assess whether injection molding could be carried out using the system. Following this success, we scaled up the machine to a 1 gram scale, pilot scale injection molder. And using this system, we can actually create microneedle patches for drug or vaccine delivery. Injection molding is a very promising method for these types of devices because injection molding is a very promising method for producing these devices because it is a solvent-free process. And using solvent-free processing allows devices manufactured in this way to free themselves from the cold chain. And if we have a dissolvable self-administrable microneedle patch, this reduces reliance on healthcare professionals for the delivery of vaccines to remote or developing areas. So the idea here is to have a conventionally machined steel plate to withstand the pressures involved in injection molding, and then have a additively manufacturable insert which fits into this plate to impart the final geometric structure. We can also use epoxy cast inserts to achieve a bit higher geometric fidelity. So using this epoxy casting procedure, we can make fairly high fidelity reproductions of these sharp microneedle devices in dissolving polymers. So here's the control sample. This is the positive microneedle mold cast in EMA. We can make a negative of that into a high temperature epoxy and then injection mold that with polycaprolactone. Here's what that looks like. You can see still relatively sharp tips, very good fidelity. We can then transition to a dissolvable polymer such as polyethylene glycol, and you can see still a fair bit of, these are still fairly close to the original tips. There is a bit of dulling, but otherwise fairly good geometric reproduction. We can also embed microparticle implants into these microneedle tips using the injection molding process. These microneedle or microparticles can be used for controlled release of various drugs or nanoparticles. In this case, these microparticles were loaded with palladium for electron dispersion spectroscopy to verify that they are embedded in the tips. They were loaded with rhodamine B, which is a red fluorescent dye for fluorescence imaging. The backing of the micro needle patches was loaded with FITC, which is a green fluorescent dye. And using these dye-loaded microneedle patches, we conducted a porcine skin assay. In other words, just push the patch into a bit of pig skin, and parts of the microneedles dissolve into the porcine skin. And you can see the results. We're achieving 200 to 400 microns of penetration into the porcine skin, and these microneedle patches are depositing their microparticle payload into the simulated skin. So what does this mean? Well, it means that we've successfully produced a microneedle patch fabrication process, which is potentially adaptable by future researchers. And the future work in this area is optimization of these microneedle patches to be a little bit sharper and a little bit lower shear rate, because viral nanoparticles are very sensitive to high shear rates, especially at elevated temperatures. So if we can reduce that shear rate down, we can really improve the applications of this method. And then finally, in vivo demonstration of this method of production of microneedle patches for the administration of vaccines or small molecule drug delivery. So lastly, I want to discuss my most ambitious project, which is a foray into the use of biological systems to make additively manufactured structures. In other words, additive manufacturing from the bottom up. So this picture here is a photograph of fossilized stromatolites. Stromatolites were some of the first organisms on this planet, arising roughly 1 to 4 billion years ago. They were an early photosynthetic organism which used phototaxis to increase their photosynthetic yield, each layer climbing on top of the previous for access to sunlight. And these formed these large meter-scale structures over the course of thousands or even millions of years. So the question now is, knowing what we know about bacteria, knowing what we know about their self-replication capabilities, within the context of additive manufacturing, how do the theoretical limitations of bottom-up manufacturing processes compare with that of top-down? And this was really the goal of some of my theoretical work. We know that the number of bacteria at a given time can be theoretically modeled by the doubling rate of bacteria and the original number in a colony. It's a very simple logarithmic growth function. We can, however, apply the exact same geometric parameters that we set up earlier for top-down systems to a self-replicating system model to create this new relationship, which theoretically describes the growth of a bottom-up additive manufactured structure, which is related to the size of the bacteria which is replicating, the doubling time, the original number, and the geometric constant. So using this theoretical relationship, what does this imply about the possible best case scenario for how long it takes to produce a structure with a given resolution? So if we want to produce a meter scale structure with 5 micron feature size, how long would that take with a hypothetical system? Well, using an FDM system, which is a single point of deposition moving along a vector, this would take roughly 34,000 years to produce a 1 meter scale structure with this resolution. Using a highly parallelized process such as CLIP with a 200,000 by 200,000 pixel array with 5 micron pixels, it would take roughly 100 hours to produce such a structure. Using a bottom-up self-replicating paradigm of manufacturing, it would take under a day to produce the structure, right? So this is all very theoretical, but what are the real advantages of using a bottom-up method of manufacturing? Well, this is the Bogla Solar Park. It's the largest man-made photovoltaic solar power station in the world. It encompasses roughly 160 square kilometers, and it produces roughly 2.7 gigawatts of electrical energy at a cost of several $billion. This is an algal bloom on Lake Erie. It has a surface area of over 25,000 kilometers, and if it was 1% efficient at harvesting the sun's energy, it would produce upwards of 250 gigawatts at no cost. Now, the cyanobacterial production of electricity is a well-studied subject field. There's a lot of work being done in this area. But what's less well-studied is the use of the ability to transport and harvest this electricity using a bottom-up method, right? So for this, we need an organic conductive material that can be synthesized from the environment in a bottom-up process. To fill this role, we can look towards the field of conductive polymers. So polyaniline is a well-known conductive polymer, and there's metabolic pathways proposed for the production of aniline from a bacterial organism. There's also a known enzymatic pathway for the synthesis of polyaniline, which is a conductive polymer, using an enzymatic catalyst and hydrogen peroxide as a substrate for its catalyst. Furthermore, there are peroxidase enzymes which have known synthetic pathways from bacteria, and there are species of cyanobacteria that produce hydrogen peroxide in response to high light intensities. So what does this mean? Well, using this method, we can potentially create large-scale structures using conductive polyaniline using only self-replicating life forms and environmental resources. Such large-scale structures could revolutionize energy capture and storage in the future. Now, the metabolic engineering of aniline production is a bit beyond the scope of this work, but the aims here are to take the first steps toward a self-replicating paradigm of conductive polymer synthesis. So firstly, I'd like to take a look at the optimization of hydrogen peroxide production from cyanobacteria in response to light. So to do this, we constructed a uniform light exposure chamber. This has a number of high power grow light emitters and a heat sink under the well plate to keep things cool so the cells don't die off too early. Next, we began evaluating the wild type Synechococcus PCC 7942 and its production of hydrogen peroxide in response to various light intensities. Now, this is an overly simplified model of the biochemical processes involved with the production of hydrogen peroxide. And through some prior testing, we know that it takes roughly 500 micromolars of hydrogen peroxide to really kick off the production of polyaniline in any measurable quantities. So with the wild type 7942, we're only getting around 2 to 3 micromolars of hydrogen peroxide, even with very high light intensities. So we really need to pump these numbers up. And one way we can do that is through the use of electron transport chain inhibitors. Now, these are chemicals that are traditionally used as herbicides or algicides, which block various metabolic pathways in photosystem 1 or photosystem 2 within the cyanobacteria. These disrupt the cellular pathway of converting light into energy and instead direct some of this light energy into the production of reactive oxidative species, which are then dismutized by superoxide dismutase into hydrogen peroxide. Now, as I said, these are herbicides, so if you use too much of them, it's just going to kill the cells. But if you use low concentrations, you can theoretically create higher amounts of reactive oxidative species without fully killing the cells. So we tried this, and here's the production of hydrogen peroxide when you use different electron transport chain inhibitors. Unfortunately, while in theory we are producing more hydrogen peroxide, there's a secondary protective mechanism within the cells called catalase peroxidase, which as soon as this hydrogen peroxide is being produced, it just gets reduced back down to water. So we need to take it one step further, which is to knock out this secondary protective enzyme. So thankfully, the 7942 species of cyanobacteria is a well-studied strain. It has a full genome mapping, and the Golden Lab has a cyanobacter tool with a number of genetic cassettes which can knock out a number of parts within the cyanobacterial genome. So we used one of these vectors to knock out the catalase oxidase producing genes. And this colony was actually stable. We can grow it up using a thiosulfate-enriched media, which is a reducing enzyme which takes care of these harmful peroxides during production, during growth. And then we wash away the thiosulfate prior to testing. So once again, this is the wild type 7942 with electron transport chain inhibitors. Really not a whole lot of appreciable peroxide production. Once we knock out this catalase peroxidase enzyme, we see 2 orders of magnitude increase in the hydrogen peroxide production. And using a bit more of an aggressive electron transport chain inhibitor, we can increase this production up to 1500 micromolar, which is well above the threshold necessary to produce polyaniline. So next we want to take a look at, can we insert the genes necessary for producing this enzymatic catalyst into the cyanobacteria? So there are known pathways for doing this. In 2003, famously, Francis Arnold conducted a directed evolution study in order to adapt horseradish peroxidase for expression in an E. coli. Now, we attempted to replicate these results, but unfortunately, it has a relatively low activity in E. coli, and we weren't able to get any significant activity. Furthermore, the expression of horseradish peroxidase from E. coli requires the incubation with extracellular hemin. So this is not ideal. We tried an alternate peroxidase enzyme called Apex-2, which is a ascorbate picoperoxidase, which doesn't require incubation with extracellular heme. This has very high activity when expressed from E. coli, but unfortunately, it doesn't have sufficient electrochemical oxidation potential to catalyze the production of aniline, or polyaniline, rather. Further research involved trying to insert these cassettes into 7942. These were also unfortunately unsuccessful. So stepping away from this, we tried next to use extracellular horseradish peroxidase and extracellular aniline and produce polyaniline in response to light. So if you put all these components together and use the wild type PCC 7942 with aniline, horseradish peroxidase, and just expose them to light, you do see some polyaniline production. This is a negative control sample where it was not exposed to light, it was just left in the dark. And then sample A was a positive control sample where hydrogen peroxide was added extracellularly. Furthermore, if you use the cocoa cells and electron transport chain inhibitors, we can see a 200 to 300% increase in the production of polyaniline just in response to light. So future work on this area, obviously there's a lot to do, but we've taken some promising first initial steps. Obviously we want to do metabolic engineering of aniline into a cyanobacterial chromosome, the incorporation of peroxidase enzymes successfully into the chromosome, and then enhancement of the hydrogen peroxide in the absence of electron transport chain inhibitors, because it's not ideal to require the presence of an herbicide in order to grow up these specialized bacterial colonies. So in conclusion, we've taken a look at improving the novel materials for top-down additive manufacturing through the use of isotropically expanding foams. We've come up with some novel methods for utilizing subtractively machined injection molding insert molds with additively manufactured inserts for the production of solvent-free biomaterial devices. And we've explored some novel methods for harnessing the self-replicating organisms to create large-scale polymer structures in a controllable fashion. So finally, I'd like to acknowledge all my collaborators, co-authors, the Corsi Lab, Hire, Golden Lab, John, the Enlace program, and the NSF MERSEC, friends, family, colleagues, business partners, collaborators. Thank you all so much. It's been a journey. Thank you.
All right, excellent talk, young man.
Thank you, sir.
Someone who's asleep on the pool table.
Oh, that's Jack, one of my business partners.
Anywho, so for the audience members that are not part of the committee, this is your last chance to interrogate David before he becomes a doctor. Folks that are in the Zoom chat, if you have questions too, I'll be monitoring too, and I can pose them, or you can unmute and say whatever you like. So questions, please. Okay, just call on people. Matt, you have a question?
Yeah, so what kind of 3D printing technology do you think would be required to print in outer space?
It's a good question. I think most 3D printing technologies you can really use in outer space, but I really think the real advantage of outer space is going to be in semiconductor manufacturing. That would be my guess, because semiconductor manufacturing, you need very high vacuums. And if you could do semiconductor manufacturing in space, you would enable a lot more possibilities. It's not really 3D printing, but I think that's going to be the most promising avenue of additive manufacturing. manufacturing in space.
Thanks, Matt. Of course. Say, anyone else?
So the synthesis of polyaniline generally requires acidic condition and ammonium persulfate as initiated.
Mhm.
So if you utilize biological method to synthesis polyaniline, does it still require that acidic condition?
It does require a slightly acidic condition. You can do this using a buffer solution. It'll still produce polyaniline even in neutral or basic conditions, but it won't be the conductive form. It converts to a non-conductive type of polyaniline. That being said, the enzymatic production is a lot faster than using APS. With APS, you mix the hydrochloric acid in there and the APS, and it takes 5 or 10 minutes for the polyaniline to really get going. With enzymatic, it's instant. It'll produce polyaniline using this enzymatic process in about 8 seconds. So, advantages and drawbacks.
All right, well, if nobody else, I don't see anybody in the chat. Let's thank David one more time, and then. All right, if folks in the Zoom could please log out, other than, of course, Professor Chad.
All right, thanks everybody. Thanks. Thanks.